of pharmacy to keep FDP.
Correct, madam. Correct. Actually, since the program was scheduled in the afternoon, many of the speakers, we have still communicated to all of them who were eager enough to listen to you. They'll be joining soon.
Yeah, it's okay. I think because there were so many, webinars and FDPs and everything. So it's okay. I think if they understand and if they can convey to others, and anyhow you'll be sharing the video.
Yes, yes.
So
yes.
they must, if they get some idea and if they are able to do something, that is the purpose of any FDP, I think. Yeah.
Okay.
If they can get some idea and do some research based on that, that will be wonderful.
Yes.
So, shall I share my screen?
One minute. I will just in the settings. Yeah, you can share your screen, madam. We'll start with the introduction part. We'll wait for another 5 minutes since we have given time to the delegates. Sure. 11:30 will exactly start.
Yeah.
So we'll start with the program, madam.
Okay.
Once again, welcome all the delegates, and I welcome Dr. Harsha Kharkwal, madam, Director, MET Institute of Phytochemistry and Phytomedicine, MET University, Noida. The topic of our session is challenges and opportunities in natural drugs and insight. So it is a very blissful moment to have her on board of the speakers of this FDP. So I wish all the delegates will have a very knowledgeful experience and presentation from this topic. So I request Professor Dr. K. Sakhi madam to introduce the guest and the speaker to all the delegates, please.
It gives me an immense pleasure to introduce today's speaker, Dr. Harsha Kharkwal, Director, Amity Institute of Phytochemistry and Phytomedicine. Dr. Harsha Kharkwal is having 20 years of experience in research from leading research institutes, having researched in natural product chemistry, isolation, characterization, leads in drug discovery, pharmaceutical formulation, plant hydrocolloids, derivatization, etc. Her academic history includes she has completed her BSc in the year 1996 from Osmania University, Hyderabad in microbiology, zoology, and chemistry. Has completed her MSc in the year 1998 from Kumaon University, Nainital in organic chemistry, and PhD in the year 2004, National Botanical Research Institute in organic chemistry. She is working with Amity University since October 2005. She has done significant research work in the field of applied carbohydrate and natural product chemistry. To her credit, she has around 45 publications in research journals, and she has 24 published chapters in various books and one published book, and she has filed around 68 patents, out of which 63 have been published and 14 have been granted. She has completed 4 projects, of which 3 are ongoing, and she has also completed 2 industrial projects. She is a mentorship for PhD also, in which she has completed 4 and ongoing are 2 PhD projects. She has also been awarded with various awards and fellowship, to name a few. She has been awarded with Senior Research Fellowship by CSIR in the year 2003, International Travel Grant by DPT and CSIR to attend the 11th Hydrocolloid Conference, which was held at Purdue University, USA in the year 2012. Has also been awarded with Lucid Research Award from Lucid Colloids for her work on gums and hydrocolloids in the year December 2004. She has also achieved top 20th position in IMC Innovative Awards in the year 2013. She is the export reviewer for BIG and BIRAC. She's also a board of mentor for AIC Prestige Inspire Foundation, which has been supported by Atal Innovation Mission, Niti Aayog, Government of India. She is a mentor on board under BIRAC BRTC program. She is also an expert in chemistry for preparation of differential dictionary chemistry by Commission for Scientific and Technical Terminologies, MHRD, Higher Education Department, Government of India. She is also a resource person for CME program for All India Institute of Ayurveda, Ayush. So I welcome you, Dr. Harsha Kharkwal, madam, and I hand over the session for you to speak on the topic, challenges and opportunities in natural drugs and insight.
Yeah. So thank you so much. Thank you again for this, and I was listening a part to the last session in which everyone was talking about the stressful life and happiness. I think hope is a decision by all of us. And I'm thankful to Kulkarni sir for recommending my name. He's there in the, I can see him. So first I'll give you a glimpse, the flow of my presentation will be, first I'll talk a little about, the university, 2-3 slides and the work that we are doing. And because it's COVID times and the future also, there will be so many mutations of this virus. So, what should be the approach, the opportunities of natural drugs in this, and also on reverse pharmacology, on different opportunities which are available in future for natural drugs, and also the challenges. So in challenges also there are opportunities. Because of challenges, we know what is the challenge, and that's why we get the opportunity itself. And also relating it to few authors, like how they have assessed this part of challenges and opportunities in natural drugs. As I also feel that, as you know, education is lifelong. So as long as we live, healthcare is very important. Good health is important for a person to live joyfully and happily. And the secret to success is to take one day at a time. But I think one should be very oriented, and should have that zeal for work should never die. And I think that keeps you going no matter what, no matter what your age, and whatever it is. Like, if you have that same enthusiasm you had as a child for science, then I think life itself will become enjoyable, and every day there will be something new to do. So plenty of opportunities in natural drugs. So I just wanted to, though you all know, you must be knowing, but I just wanted to introduce our university also, that we have around 10 universities in India, and we have overseas campuses also. There are more than, 6,000 faculty, more than 150,000 students. There are more than 150 plus institutions, you name it, they have it. And we have got very good infrastructure, and it has been benchmarked against the best universities around the world. So the picture is actual picture. It is not something factual. It is the space that you can see in, like we have all over India. We have many management institutes also across in overseas, and in New York, there is a whole campus, Longway Island. I just wanted to give you, so the scientific discipline, it has a strong focus on research. It is a research and innovation based university. Just yesterday, 4 technologies of food technologies were transferred to an industry. So the stress is on biotechnology, food technology, nanotechnology, herbal, microbial, pharmacy, engineering. So we have got good labs of pharmacy. We have got good labs, and also in biotech. Yeah. And we have a very good law college also. So we have Amity Science, Technology and Innovation Foundation, which gives the main focus is to promote science, technology and innovation in all Amity institutes, and to promote cutting edge research. And we also have lecture series from eminent scientists, from Nobel laureates also. And we have internal project funding, as well as we have a system where the projects are being screened, and finally it goes into the final evaluation. So Amity has got more than 1,000 research papers published in leading journals like Lancet, Nature, scientific reports. And there are many case studies which are developed. Center of Excellence is there. Many Ramalingam fellows are there in our university. And talking about my institute, like, it's a very small institute, but we are very focused. We only have PhD program, and we take dissertation students, and we could transfer technologies, and we are still in pipeline to transfer more technologies. And uh, this was an old data, and right now, like, there are 14 granted patents. So all the patents which were filed from Amity Institute of our department, like phytochemistry and phytomedicine, were from indigenous sources, and they are of commercial utility. So this is a granted patent, and this technology has been used in one of the industries in Jaipur. And in this, we could carry out the toxicology studies also in animals. So it can be used both for, and one of the industries, it went about to see its scope in 24 European countries, and it was found to be very good. So because in Europe there are very stringent laws, standards which are being set. So this was one of the patents. So I also wanted to stress that in natural drugs, one of the constraints, major constraint, is TKDL guideline, which comes when you are trying to patent anything. Because if you see, India is rich in its, India is known for its traditional knowledge, yeah, and the kind of flora and fauna that we have got rich wealth, rich biodiversity is there. But what lacks, to really move ahead in future is one of the blockages is TKDL guideline. Like, if one species is known for one particular activity, then the whole family, even if you want to patent that, even if you have done that for the first time, still you cannot patent it, because uh, one of the species is known for such kind of activity. So what is important is that to go, how can we go for breakthrough, how can we cut through that, and still prove, still are able to um, uh, bring out the research by utilizing the resources. How can we do that? So we have to go mainly, this is also a granted patent, and this is a self-defense spray, and it has got longer period of irritation time, and almost there is temporary blindness for whole day. And this is also from a natural source. We prepared a semi-synthetic derivative. So by value addition, one of the parameters by which we can utilize, or we have opportunity in future in natural drugs also, is to utilize the drug for its activity by in making interventions in that. So the intervention can be by going for derivatives, by nanotechnology, by reverse pharmacology, by various knowledge, by combining all the knowledges, all the knowledge that we have. So this was one of the other patent, which is a granted patent. I'm just showing what are the related patents related to natural drugs. Then we have this was appreciated by Ministry of HRD also. Like, we have vegetarian heart capsules, and we have got 2 incremental patents in this. In this, it's purely because you are all are from pharmaceutics, and this is purely a gelatin-free composition, and it is prepared by utilizing, polysaccharides. Some are novel also, which are used for the first time. But uh, the only way which I have shown is by their synergistic formulation, by showing them to be more economic. And this is also a granted patent, which was granted by, Indian Patent Office. So we carried out all the tests, the quality control parameters, which were the requisite for any, patent. I feel, before publication, if we can file a patent for any good idea that we have. So nothing is small or big. Yeah, an idea is an idea. Maybe, there are many capsules which are gelatin based, but we just thought, and this was because world is moving towards organic. Yeah, and only thing which is required is like how you are able to do all the quality control parameters by NABL accredited labs. So the results which are required by any authority to authenticate. How will you authenticate? Because in this is a lab grade sample, like, but it is a granted patent, but I could get the results from NABL accredited lab. So this is where we have Technology Enabling Center in Amity campus, where I got the funding for enabling my technology to another level so that I get a patent, a granted patent. Otherwise, that idea will remain somewhere, in the desk. So this is also a granted patent, and this can be used for childhood diarrhea, but this is one of the areas which requires more thorough investigation, because what test on the basis of which it was granted was that the composition showed significant activity against E. coli. So, and the the normally whatever we have chosen, it had a good literature backup also, plus we could carry out all the tests we could give, because after though you all may be knowing, but I'm just telling, after we file a patent, like it takes 18 months for publication, and then it is on public domain, and then we have questions. First examination report comes and we have to answer all the questions, and still if the examiner is not satisfied, then we have a hearing, and in the hearing also we have to defend the work what we have done. So this patent which was granted was based on the test which was carried out to show that these have good effect against any gastrointestinal infections, and how each of them have a role, so by toning the lining of small intestine as well as enhancing the growth of right bacteria. So we could give the traditional results, traditional knowledge results that we have in the form of all the literature, as well as doing it actually in practicals, like what was there to prove that it was non-toxic, to prove that it has got an, it can be used against diarrhea, and it is safe. It is cost effective, which has balanced composition of electrolytes, and it maintains optimum pH of digestive tract. So like, and this is another granted patent on diabetes, where we have, this is a unique fiber blend of carbohydrates, and it is fortified with suitable vitamins, minerals, and whey protein, and it provides the required energy intake. Okay, and this can be also utilized, like we have made tablets and all, and one of the doctors also tried it on their patients, like they had taken, they had funded us for preparing a particular formulation in the form of sachet and tablets, and also for all the initial raw materials, because the cost of raw materials for all this is less than rupees 100, so, and it is hardly like 1 gram, which was very effective, and they could try it on patients also. So we could support it with the lab-based data, as well as based on human trials, because it was safe to use. Like we can eat tulsi just like that. We could prove that, this formulation can be also taken. So once taken, we could see. So the other thing that we did in our lab is we work on many, like we have projects on medicinal plants also. So one of the interventions that we did was utilization of them to prepare nanoparticles. So here, by enhancing, by we are using mostly the ethanolic extract or the water extract of the plant, and even with 1 gram, if you have your substance, pure substance, or even with 100 milligrams, you can prepare the nanoparticles and see the activity. So here we prepared silver nanoparticles. The color change will show. So we normally go for, so this is one of the interventions. It is also, it can be used for, as an opportunity in future for natural drugs also, because it will enhance the activity. Yeah, so we characterized the sample, we carried out the SEM, the FTAR, the DLS analysis. Okay, and so if you get silver nanoparticles show a peak at 430 nanometers. So if you get a peak at 430 nanometers, that means your nanoparticles have been formed. So we have the requisite facilities here in nanotechnology lab, where we prepare our nanoparticles like zinc nanoparticles, gold, silver. So it will also enhance the solubility of the drug. It will, there are bioavailability problems with many drugs, because of which we are not able to use them. Yeah. So, so intervention of nanotechnology in that sense can be researched in that way. Yeah. So this was one of the research that we carried out. We also carried out the antibacterial study. Another thing which we carry out in our lab is bioactivity guided fractionation for isolation of bioactive compounds which act as lead in drug discovery. So in this, because you get, yesterday I just wanted to share that, if we see this flow chart, yeah, like there are different schemes. By different schemes, you can isolate different compounds. Like my student, when she did it by this method, she could isolate 9 novel compounds. Like the compounds have been known, but from the lesser known fractions of, medicinal plants, she could uh, isolate 9 novel compounds. And then uh, we know the procedure. Yesterday, somebody was asking about MALDI-TOF. So I wanted to say that it is matrix assisted, fragmentation technique. So we know that mass spectroscopy is a fragmentation technique by which you know the molecular weight of the compound. And you know the molecular weight up to the decimal point by using mass spectroscopy, and that is why it is such a important technique when we come to when we come for isolation and characterization. So we all know the steps. I just want to just go through it once, like how do we go about it. Once we get the raw material, we extract it, we do the TLC, we concentrate it, we again do the TLC, we see the space between the spots, the RF value, if it is at a the distance should be large enough so that we can go for column chromatography. So it can be either normal, which is from non-polar to polar, or it can be reverse, where it is from polar to non-polar. And by the elutes that you get, you again go for TLC, then you go for UV, you go for IR to know the number of the kind of functional group presence in that, you go for HPLC to by the help of retention time, by taking the standard drug, you will know, okay, this drug is present. And so you have different techniques. Like then the purification is done. After that, you go for molecular weight analysis by mass spectroscopy. So in mass spectroscopy, normally the fragmentation is done within seconds, but in MALDI, a matrix is made of glycerol. So the compound and all these techniques, whether it is mass, NMR or X-ray, you need pure compounds. Otherwise, there will be a lot of noise. Like in NMR, there will be many small signals. It shows the presence of impurity. So when we are going into natural products or we want to do something, so we have to be at least thorough in theory as well as in practicals when we are doing it. So we must know that, there should be no adulteration of any kind when you're handling the pure sample also. So if you see the in matrix assisted, what happens? You dissolve the compound normally, like in glycerol, in any viscous solvent. So your drug is entrapped. That viscous solvent will form a layer around the drug. And normally xenon atoms are used, which have having high translational energy for the fragmentation of the drug molecule. So normally the fragmentation which takes place within seconds, in case of matrix assisted, it takes place in half an hour. Yeah, it takes place slowly so that the proteins, the polysaccharide, because it is used for high weight molecules, it is not getting degraded, as well as it is getting fragmented. So you go reverse. Once it is fragmented, you I have shown in one of the slides that how. So we carried out this methodology for one of our projects, like these are the fingerprints from HPTLC. So you see also the variation in different areas from which the sample is collected. So this was a pure compound which was isolated. So this is how we will we report when we isolate a drug, natural drug. We give all the data, like right from mass to NMR, and this is how the fragmentation occurs. Yeah, you can see the fragments, the lines which are given. So when you combine all these fragments, you will come to know the molecular weight. Similarly, for 1H NMR, you will know the number and nature of protons, the chemical nature of protons. Similarly, for 13C, you will come to know number and nature of carbon atoms. And because all the most of the organic molecules, they contain carbon and hydrogen, and that's why these techniques hold such importance. And in X-ray diffraction is a very important technique. If the sample itself crystallizes, or you can crystallize it by making a supersaturated solution. Normally, by making a supersaturated solution in the drug in which it is soluble, and then you have to add the solvent in which it is not soluble, and keep it for few days. You will see the crystals will form as well as they will grow. So if you sometimes go to CDRI, Central Drug Research Institute, you will see the whole building is there for X-ray crystallography. And we had one Subramaniam sir at that time, and he was very strict, and they used to do X-ray crystallography of proteins. So talking about the opportunities of, in natural products, one of them being because we all have gone through COVID, and that is why we are facing this kind of stress also. So one of them is the bottlenecks for antiviral drug developments. What is it that we require, So availability of libraries of plant extract based on sustainable resources. Yeah, we can utilize them. Need for fast antiviral screening system, both in vivo, in vitro, screening in low and high therapeutic environment, then using cultivated plants or microorganisms suitable for large scale fermentation, collaboration with specialized laboratories. So if I'm not wrong, BSL 4 level lab is also available with Baba Ramdev's lab have this facility, and develop. That's why they were able to come up with the COVID drug and develop pre-selection methods by using metabolics guided fractionation instead of bioassays. So normally we use bioassays for selection. So we can go for metabolomics guided. So approaches to corona drugs. This is just an how we can approach. So we know repurposing of drugs in that. You don't have to register. It is easy to register, and the specific, just 1 second. Yeah. So the specific, treatment of symptoms can be done by going for repurposing of drugs. And when we look out into compound, this is the approach that we must make when we are doing, because if you see right now, most of the projects are being funded for COVID right now also, because there's so much of mutation. The third wave is also here and there coming. In US every day, 1 lakh cases are reported. But like So we have to be still, we need to be very careful about this COVID. We cannot take it for granted. And as well as because it's a challenge, it gives you an opportunity that how you can go about it. So then there are biologic extract libraries, the bottlenecks being you have to identify the actives and also scale them up. And advantages is that with extracts, you can go for elaborate development. Same with libraries. And if you have known antiviral extracts, like this is one of the, antiviral drug, Swasth Rakshak, given by All India Institute of Ayurveda, which contains all the medicinal plants. Yeah. And it is used for proper breathing. Yeah. Then we can also utilize the traditional medicines for infectious diseases. So advantages is that it's a complex mixture, so there is a lot of synergy. Yeah. And there can be, because there is an evidence based on traditional role that they have, it can be traditional based clinical trials can be done to establish to go for further for corona drugs. So as such, we can always look out for repurposing also for known antiviral extracts, because in some cases, the drugs which were used for SARS were useful for COVID also. So how utilizing our the drugs which are available within India, our indigenous sources, we can approach for corona drugs. This can also be one of the approaches for opportunities which COVID has provided in this time for natural drugs. Okay, so this is from one of the papers that Vaidya sir has written, like where he has said that herbal drugs, they constitute a major share of the officially recognized system of health that we know, whether it's Ayurveda, Yoga, Unani, and more than 70% of India's 1.1 billion population is still using non-allopathic system of medicine. So currently, though there is no separate category for herbal drugs or dietary supplement as per as Indian Drugs Act, but it again offers immense opportunities for observational therapeutics and reverse pharmacology. And we can have evidence-based herbals are used in the diverse system and manufactured as per the pharmacopoeial guidelines by a well-organized industry. So yeah. So in reverse pharmacology, as we know, there is yeah. So in reverse pharmacology, as we know, there is experimental documentation. Yeah. And then there are studies, experimental research, and clinical phase 2 trials, which leads to the natural drug formation. And when you isolate, suppose, active principles from the Ayurvedic therapeutics, which are already known from the medicinal plants, and you provide a lead, then it leads to formation of a modern drug. So we all know that, yeah. But this provides an opportunity, especially when we're using different drugs. So I'll be also sharing a slide on, future healthcare. In that, what are those diseases in which already we have certain plants which are known in nature against those diseases, and how can we explore them further? What opportunities do they provide? So for any, you know, any drug that we are approaching in future for an opportunity, it has to be thoroughly investigated. Yeah. And that can be done only through proper preclinical and clinical trials that we can come to a conclusion. Yeah. So we know that India is having a pluralistic healthcare system, and it offers an unfettered choice for the quest of new clinical effects of traditionally used medicinal plants. And it was one of the scientists, Chaudhary Roy, who had given um, a neologism for such a discipline. He called it as observational therapeutics. And he also expressed his hope that further research directed at few of the chronic disease against which more drugs are needed, such as diabetes, bronchial asthma, could lead to the discovery of new drugs for these conditions. So observational therapeutics is one which traditionally it has been used, and we are observing what are the therapeutic uses of these. So if you see, the drugs have been categorized, whether it is phyllanthus, it has been categorized as for safe usage. Okay, ashwagandha, it comes under dietary supplement. So all those which comes under dietary supplement and which are not under IND, or which are not undergoing IND, can still be researched for new drug, natural drug applications. So phyllanthus, which is known for anti-aging, which is known for safe usage, needs to be thoroughly needs to be thoroughly proven for its efficacy by proper undergoing proper clinical trials. In the same way, one which is considered to be a dietary supplement, if it is non-toxic in nature also, and if it can, but still when you are preparing a formulation, you have to see whether if there is anything. So these all drugs which are there in plenty in India and have been known to be Ayurvedic drugs, are still not used to that extent, and maybe they have other uses also. Suppose if some out of them, like we know Brahmi has got very high antioxidant value, so beside cognition it can be used for cardiovascular diseases, for all these, and it is evidence based. But still, a particular plant species which is known for only one particular activity still needs to be researched for other kind of ailments to prove its efficacy. So this also provides an opportunity, mm, because we have got a system in which at least the status has been marked by National Medicinal Plant Board, by Ayush. They have given the status by All India Institute of Ayurveda. Dr. Tanuja Nesari ma'am is there. So we can always, she's like an encyclopedia in case of Ayurveda, and but the only and they were providing for COVID patients, for all the for defense people, they also provided. That was also one of the centers, All India Institute of Ayurveda here in Sarita Vihar, and many of the patients got cured by using their formulations which consist of these drugs. But still, for its wide acceptance, it has to be investigated further. Unless it is investigated, it will have a limited number of approach. So this also provides an opportunity. And we know that since ancient times, many Indian medicinal plants have been used globally. For instance, Indian aloe vera, it is known as Grit Kumari, and we know that right from head to the toe, it has got multiple uses, and it is known for its anti-aging effects also. But still, that anti-aging effect part has to be researched. Similarly, the plant Adhota vasica has been extensively studied for cough, and however, their potential for use in bleeding disorders and tuberculosis remains untapped. So how can we, when we are trying, and you see the international projects which one gets is by having collaboration with such kind of drugs only, which are already known for some kind of effect, have been proven for certain kind of activity, but the other disorders have not been thoroughly sought for. So in that we need collaboration, and only by collaborating, you know you will come to know, you'll be able to utilize it. So reverse pharmacology may lead not only to the new leads and drug candidates, but also to novel targets and pharmacodynamic efforts. So for example, Coleus forcefully, we know forskolin has multifaceted efforts, and but uh, only after it was mediated by activation of adenylate cyclase and increased concentration of cyclic adenosine monophosphate, it has been served as an important tool in molecular pharmacology and endocrinology. So the complexity of effects of even simple phytomolecules can be done in future. So the emphasis has to be primarily on the human effect. So whatever human effect that you are getting, though it may be known for any kind of ailment, but the emphasis which the Roy Chaudhary had stressed, one of the scientists, for opportunity, is observational therapeutics. By observational therapeutics, we can move ahead for new leads and drug candidates. Okay. So, in general, otherwise, if you see, okay, one of the slide is missing. Yeah, I think I've deleted that slide by mistake. In that, what are the opportunities as such for in the area of natural drugs or natural products? Is that one is definitely because most of the natural drugs are one which are derived from nature, so and mostly medicinal plants are used. So opportunities lies whatever in entrepreneurship, in case of if you want to go for medicinal plant cultivation, exports, in drug manufacturing companies, in teaching professions, as drug inspectors, as pharmacogonist, as Ayush practitioners, for clinical and research opportunities, and also in newer fields. So right now, the opportunities also lie in, I'll be taking in future slides also. So in this, like, we have computer aided drug design, we have metabolomics, which has been used as an opportunity, and we have AI system also, which is used against. So in AI system, only when the drug is effective, yeah, in artificial intelligence, we can utilize in case of natural products also. And ICMR is funding nicely for AI-based projects. If you have AI-based projects, artificial intelligence projects, and when we go for kinetic modeling also. So all those techniques also provide opportunities in natural drugs. And we know the challenges when we come to natural drugs lie in their standardization, the management within ranges of risk, the safety and efficacy, the constraints with clinical trial and people available. So in one of the research papers, one of the scientists have given 60 challenges that we can see in case of natural products, which I've covered in my slides. But when we are dealing with, suppose, medicinal plants as such, I had seen the presentations, few glimpses where people have covered about good manufacturing practices, like where the processing of herbs is given, and such as like how do you collect them. Then there is similarity between both the drugs, which leads to adulteration while collection of the plant species itself. So these are the challenges, but then that's why role of each and every person, right from a botanist to a taxonomist to an ethnopharmacologist to a chemist to a pharmacist, everyone has to work together to overcome such challenges which we see. But challenges are also described in detail in terms of the time process which is taken when we are going for natural product drug analysis. Yeah, other factors are there, like there are mostly adulteration issue, which is one of the main issue with the raw drugs that we get. So if you see the limitations of natural product when compared to synthetic the synthetic drugs which are available, is building up and maintaining a high quality natural product library, which requires a skill set which is not generally available in the industry. So if you can specialize in that aspect, that provides an opportunity for you in the future. And they are normally the natural drugs which are available. They are in small quantities, and the purification process is also time consuming. Yeah. So rediscovery of known compounds is also a major problem when we are going for screening natural product libraries, and because it is due to the lack of efficient dereplication methodologies for both natural product as well as the compounds in the natural product libraries. And we know that mostly the natural products, because they are combination of many APIs, it is structurally complex. So modification of complex natural product using organic chemistry is very challenging. Yeah. So but because of recent technological advances and development of new methods, they have revolutionized the screening of natural products, and thus they offer a unique opportunity to re-establish these natural products as major source of drug leads. And they can always address the limitations of screening of natural products. So the advances that are being done, like when you're combining, as you will see in future slides, like when you're combining liquid chromatography with mass spectroscopy, throughput screening, and when we are going for AI techniques or computer aided drug designing or metabolomics, which actually reduces the time, and it is very focused, and there is the screening process is also very thorough. So development of streamlined screening process of natural products, improved natural product resourcing has been done, and there is combinatorial biosynthesis and molecular microbial genomics also play a role. Okay, so this, pharmacodynamics, yeah, this is your field. So using biological assay is also one of the methods for quick identification of lead compounds, and it is rapid, it is simple, and it is not very expensive. The most important thing is it is very specific. So when we are going, active natural products have served an important source of drugs since ancient times. And still the chemodiversity in nature in plants and microorganisms, they offer a valuable source of novel lead discovery, but rapid identification of bioactive compounds remains a critical factor to ensure that this tool of drug discovery can compete with recently developed technologies such as chemical compound libraries and high throughput screening. So rapid screening, it requires availability of libraries. So one of the industry, it gives it has the library for natural compounds, which I'll be naming now, and the coupling of chromatographic methods such as high pressure liquid chromatography, mass spectrometry, NMR, with online bioactive assays. It's an important tool for high therapeutic screening of natural product mixtures, which will give rise to the lead compounds. Okay. So mostly there are four major roles of the bioassays, which are pre-screen, screens, monitors, and secondary testing. So in pre-screen, bioassays applied to a large number of initial samples to determine whether or not they have any bioactivity. So which reduces, the time limit, and they have very high capacity, low cost, and they give rapid answers. And then in the monitor, a bioassay, it is used to guide fractionation of a crude material towards isolation of pure bioactive substance. In secondary testing, lead compounds are evaluated in multiple models and test conditions to select the candidate for development towards clinical trials. And so what are the strengths, the Indian pharmaceuticals? So this is from Directorate of Medicinal and Aromatic Plant Research from Anand, where, we had a training there where they spoke about the key strengths of Indian pharmaceutical industry, which actually is the basis of the future of, natural drugs. So one is strong manufacturing base that the Indian pharmaceutical industry has. There is cost competitiveness, network of laboratories, highly trained pool of scientists and professionals, world-class quality products, then fast growing healthcare industry, rich biodiversity, and the established industries have high quality approvals from US FDA, MHRA, etc. So these are the strengths of Indian pharmaceutical industry. So when you look, at the future and opportunities, why do you want to, go for natural products or natural based drugs? And we know that natural drugs are one which are procured from nature, whether from marine source, from plants, from animals, any source. But if you see, especially the plants which are used mostly in formation of natural drugs, if you see the value and the share that India holds, so India stands sixth, and it is like 5.76%, and the value is in US million dollars. So if you see Sanat Pharmaceuticals, if you see many pharmaceuticals which are just selling extracts, but they are uh, defined extract, 95% purity, 20% purity. This is the share that India has. 57% US million dollars is there for, and India ranks fifth, and out of 79 countries, it ranks sixth. And in licorice and ginseng only, we have got like the value is worth about 272 US million dollars, and the crude drugs in total, yeah, they have 1,277 US million dollars opportunities there. So Indian strength in herbals, we all know in Indian system of medicine, medicinal plants are used by tribal communities. They are also utilized in modern Amchi, Siddha, Unani, Ayurvedic systems. And if you see the actual figures, the 80% of raw drugs come from wild. And in Gujarat, which is the hub of Ayurvedic drugs, as well as where they utilize the natural sources, even the animal source. Like if you see, they prepare those bhasm, and we could visit those industries where they utilize the bones also. So the annual consumption of raw drugs is around 3,19,500 million tons. This is the requirement. So and 134 species are cultivated and 160 are partially cultivated. And National Medicinal Plant Board actually gives funding, and it also gives subsidy when you buy pilot scale instruments, so we could get in touch with those people who were into this industry, who were preparing extracts, doing the lead compounds with the help of the funding that they got. Then export of raw materials itself, India earns around $114 million industries there. So it is estimated that, the global market will be around $5,000 million. It will rise from where it was in 2002 to $60 million to $5,000 million, because 80% depend upon traditional system of medicine of primary healthcare. The only thing which is the gap, which is a challenge, is the clinical evidence. Yeah, if you have an evidence-based study, then nothing can stop India. So China and India are the world leading exporter of medicinal and aromatic plants, and this is what which Ministry of Ayush under DMA APR is looking forward for world healthcare situation in 2050. The factors which will lead to major health problems, though when we attended this training, there was no COVID. Otherwise, I think the first major health problem would be viral infections. Yeah. And also due to pollution. Yeah. And so the factors which will constitute this are like population, food habit, climate change, lifestyle, genetic, pollution. And the major health problems which can be in which we can seek or see an opportunity in which we can go about, whether it's a traditional formulation, whatever idea that you have. Yeah, please formulate it. Please uh, see that, a final. Nowadays, the ministry is also asking, like, whatever project that you're telling, what is the outcome, what is the end result? Is there anything substantial which is coming out? So whether it is a small thing or big thing that you do, whether it is like immunity enhancement or any side effect which it is reducing. So one of the for cancer, we have got a granted patent in cancer also. So in that, we have utilized the synthetic drug which is used and combined it with natural drug, which actually led to reduction of side effects. Yeah. So by utilizing click chemistry, so Amity also has has a center for click chemistry, in which when you are trying to bind a synthetic and a natural compound which do not actually bind with each other, then you use a linker by which you link both of them. And actually by using the bioinformatics tool, by seeing what genes are being getting expressed, what are not expressed, you'll be able to see how much tumor is getting reduced. And this was done on animal model in pharmacy lab itself, and for liver cancer it was done, and there was a reduction in the tumor. So the patent was granted based on those results. But the innovative part or the intervention part was that we have combined it, the synthetic drug with the natural drug. So here, whether it is cancer, hypertension, malaria, viral dengue, or I had shown you like what can be the approach for corona drugs, which was shared by one of the scientists in one of the webinars, which I really liked, that how to go about, because the funding is mostly on COVID nowadays. So people will be utilizing both traditional and modern medicine by 2050, this was assumed, and the herbal remedies which are available, which we all know. But still, most of them, except like Artemisia annua, artemisinin, we know the Nobel laureate from China got her Nobel laureate because of this artemisinin. Yeah. So whether for hypertension, for diabetes, yeah, we have Gurmar, Memijo. So these are the plants which have been grown at Anand. Yeah. And if even if you go for cultivation, and if you see the change in secondary metabolites which are there, and if you can validate it with the help of HPLC also, then also there is an opportunity for projects, for funding. Yeah, in different climatic conditions, in different soil conditions. So we had a project in which we did it on barren land, like we could grow on sodic soils the medicinal plants, and then we isolated the secondary metabolites, established them, what was the how much percentage of secondary metabolites are there, and we got funding, and it was extended for 2 years more because of the results that we could procure. Yeah, so this also provides an opportunity. All these which we know, like um, we have in our labs, senior people who are working on like muconapurins. We have a good team which works on all this. So we have a project in which we are working on dementia, effect of dementia by using Vidania. Yeah, effect on dementia. So though we know that it is used, but by synergistic combinations, we are trying to formulate a novel formulation by comparing it with other uh, we do a comparison with the drugs that you get in the market from Unani formulation, from Ayush formulations, from different formulations that you get in the market with our formulation. So if there is a enhancement in the activity, yeah, by doing all the tests which are required, so it's very clear, like you have to know the pre-quality control parameters, the post-quality control parameters that have to be carried out, and all the steps. So the up to pre-clinical trials, we establish any kind of research that we do, we establish it up to that level, and then only we say that it is useful, and then only it's proven. So these are the common drugs which we all know, which are available, whether it is morphine, whether it is plaxitacil, digitoxin, vinblastin. So from these plants, suppose we have utilized the roots of the plants. So now let us utilize the leaves and other plants, the lesser known sources. It will also have similar kind of activity in other activities also, related activities, to find an opportunity. So there are around 1.5 million practitioners of Indian system of medicine, which are using medicinal plants in for curative applications. And there are many herbal industries, and which cater about 12% of global herbal trade, and we utilize different parts of the plants, and the most uh, the majorly we utilize the roots of the plants. 30% of the formulations are the roots which are being utilized. So so we know that medicines, this was by a paper by Jackaloff Itil in 2020, where he also said, like, medicines derived from natural sources, they make about half of the prescription medicines, and they are likely to do so in the future also. Yeah, so the importance of natural drugs in modern medicine is not just a history, but they are an excellent source of chemically diverse lead structures for discovery of new drugs. Yeah, and so the topic of his paper was also, this was by Sanjay and Saklani in 2007, but it is a good paper where they have spoken about the challenges and opportunities in drug discovery, and which involves a multidisciplinary approach. We know that, whether it's combining botanical, ethnobotanical, phytochemical, and biological techniques. So we have got new chemical entities for development of drug against various pharmacological targets, including cancer, AIDS, Alzheimer's disease, and pain. And several natural products of plant origin are in clinical use, including, we know, plaxitexil is there, art ether is there, tiotopium, to name a few. And some are undergoing phase 2 and phase 3 clinical trials. So although they continue to provide the important source of new drug leads, and also they give a plenty of opportunity, right from entrepreneurship to whether it's like if you want to establish your own industry, or you want to go for research, or you want to go into medicinal plant cultivation or extract development or lead compounds, or if you go for marine drug sources or marine sources as such for natural drugs, like we have a term called quasi drugs also, which you all must be knowing, in which we can use cosmeceuticals, nutraceuticals. So algae based projects are given, which because algae is a source of nutraceutical, and it has got a high level of antioxidants, so it is used as a food source also. So all of them provide plenty of opportunity. Whatever field you want to choose, yeah, you can choose in the terms of natural products. And for research, there is plenty of research to be done. Even if you pick up one, if you are very focused, and if you pick up viral diseases or bacterial also, there are plenty of them. The only thing is you have to remain focused on what you want to do. So although we know that they provide good leads, but the authentication is also a very important part in when you're procuring plant materials. Like in Swartia Chirata, there are two plants which look almost similar. So while collection only, if there's a mistake, then the activity will also the therapeutic activity will not be there. So an implementation of high therapeutic screening, bioassays, and scale up of bioactive lead compounds is also important. Yeah. So one of them being value addition, what we had shown, that how you can do. One is by making the derivatives semi-synthetic and synthetic. It is not like if you're taking polysaccharides also, suppose guar, that sulfated guar is used for cancer also. Yeah. So you are actually enhancing the therapeutic value by doing interventions, and by synthesizing them, by using reverse pharmacology, by thoroughly reading the literature, and then taking up the challenges which are there. So I will just go through this. So the 10 examples of the drugs which have been very popular, and which are used in our healthcare system. There are artether, there is glathamine, yeah, which was discovered from Galanthus woronki, and it is used against Alzheimer's disease, which slows the process of neurological degeneration. But in this, the opportunity lies in that when you have a parallel system of medicine, can you combine it with that? Like in case of neurological disorders, there is one system like small magnet therapy also. So when you're utilizing that kind of therapy, in which you are not taking any medication, but actually it controls the toxicity inside your body, and people have there was a reverse in their neurological disorder by overcoming this therapy of by utilizing this small magnet therapy, which is by one of the doctors, Professor Shahroz Rizvi of Allahabad, and he actually controlled gangrene also, and he has cured many people. Like one of my friend is surviving on one kidney since many years by utilizing that therapy along with the medication. So it also provides you by utilizing, by combining alternate therapies. Yeah, it gives you an opportunity how to research further. Then there is morphine, which is used for pain medication, but it should be only used for pain medication. Yeah. And then excitation is there, which is used as an anti-cancer agent. So can these anti-cancer agents be used for antiviral therapy also, that we can see? Yeah. So all these drugs which are already known, which have been utilized for you can go through this presentation, because I was thinking I'll cover the other part also. So that's fine. So Google, I think all the pharmacy people know about Google. So we all know that, what are the constraint? The main constraint is the average period. So how to reduce that average period of 10 years, normally which takes an $800 million, and out of 5,000 lead compounds, how do you get a single entity? How are you able to break through this kind of constraint? Yeah, is by actually going for observational therapeutics and seeing whether this is generally recognized as safe, whether it is having any toxicological effect, and if it has been used for human consumption, up to which level it has been used. So combining all these factors, yeah, and taking different approaches, then we can actually reduce. And definitely by having enhanced screening technique, as well as thorough spectroscopic techniques that we are using, characterization techniques, that it will lead to new drug formation, which is having high therapeutic activity. Like we are using cell lines to know how effective it will be for different ailments. Like for cancer, cell lines are there. So the preliminary test itself will reveal whether we must go ahead with it or not. So all these parameters actually help in reducing the time consumption. Yeah. So I will directly come to the challenges also. So this we all know, like how do we go about it? Like there is lead identification, optimization, and lead development when we go for a drug discovery process. And the one of the challenges in natural drugs, if it is not a pure compound, if there are crude extracts, then they are difficult to formulate in solid dosage form due to their hygroscopic nature, and they have to be masked for this property. And most of them they have poor solubility and stickiness also. So one of the I just want to, yeah. So we have utilized only 65% of flora, which has been surveyed so far. Though we are one of the 18 hotspots of plant biodiversity. So this also gives us an opportunity to thoroughly go for those kind of, plants, and also for marine sources, especially, which have been very less researched. And yeah. So globally, there is a need to build natural product extract libraries. In India, though some institutes have small plant extract libraries, they are not in public domain. So the only information which is available, it's from Nicholas Piramal India Limited, which is one of the major pharma players in India, and it has built up a plant extract library having 6,000 extracts prepared from around 2,300 plant species collected from all over the world. Yeah, so the library plays a very important role for future drug discovery, and it also offers special features in comparison when we see the natural products, natural drugs, which confer both advantages and challenges in drug discovery process. So they normally have a higher molecular mass, a larger number of sp carbon atoms, but by combining them, by synergistic combination, by uh, applying nanotechnology, we can also reduce the consumption of the amount of drug which we are using. Yeah. So this was another paper in which they have spoken about the advances and opportunities. Um, so the challenges, accessing sufficient biological material to isolate and characterize a bioactive natural product may be challenging, and although the complexity of natural product structures can be advantageous, the generation of structure analogs to explore the structure activity relationships and to optimize natural product leads can be challenging, particularly if synthetic routes are difficult for them. So if you see in this, this is an outline of bioactivity guided isolation steps in natural product drug discovery. And in this, if you are taking it from organisms or from any plant source or any natural source which are nature drugs, you're getting the crude extracts. So there are new methods for de-replication, new methods for extraction and pre-extraction, pre-fractionation of extracts can be done in this. Yeah. And the problems that the presence of natural products that are known, the presence of natural products that do not have drug-like properties, and bioactive natural products are present in insufficient amounts. So these are the problems. The solution to them is when the presence of natural products that are known, then you find a new method for de-replicating that same natural product, maybe by isolating them. You are able to get it in higher yield, yeah, or the therapeutic activity is enhanced because of the method of isolation, or you are able to isolate newer compounds by your method of isolation. And then if you have bioactive natural products which are present in insufficient amounts, then new methods of extraction and pre-fractionation of extracts can be done. If it is not possible to culture a organism out of a natural habitat, then you have to find out new methods for culturing. That can be done when you're utilizing when you're preparing cultures. Yeah, you can use different medias to procure that kind of culture which you require, or relevant natural products are not produced when the organism is taken out of the natural habitat. So what is the solution? The solution lies in new methods for synthesis, induction, and heterologous expression of biosynthetic genes. And if there are unknown molecular targets for natural products identifying with phenotypic assays, then there should be a new method for molecular mode of action elucidation. So these are the problems that we normally face, in natural drugs when we are researching it. So how do we, the challenge, how can we how can we address those methods? The challenge can be addressed by development of methods for accelerated elucidation, by seeing the drug affinity, responsive target stability, by proteolysis, by profiling, by seeing the bioinformatic based analysis can be done, because not everything we can base it on experimental data. If we introduce bioinformatic tool, then also we are able to overcome the challenges that we face. So I'll come to the challenges that we have. So yesterday metabolomics was covered. So it provides accurate information. It can be one of the tools to overcome this challenge, and to we can prioritize natural products for isolation to accelerate de-replication, and also for having new natural products. Scaffolds metabolomics can be used because it can detect the differences between the metabolites composition in various physiological states of producing organisms, and it also enables the generation of hypotheses to explain them. And it also provides extensive metabolite profiles to underpin phenotypic characterization at molecular level. Yeah, so if you want to, especially for genetic related, diseases, then you have to go for what do you say, up to the gene level, molecular level study is very much required. So suppose, for application of advanced analytical technologies empowering modern natural based drug discovery. So this is an example of how, if you are taking bacterial strains, then you can go for image type phenotypic bioactivity profiling. So here, out of 234 natural product extracts, finally 15 new metabolites were isolated by this flow chart. If you follow this chart, it is like how by applying application of liquid chromatography along with high resolution mass spectroscopic metabolomics in the screening of natural product extract, in which 234 extracts were subjected to image based phenotypic bioactivity screening, and clustering of this data resulted in the discovery of new natural products. So if you combine and use a metabolomics tool, then you can screen out the compounds depending upon the molecular weight of the compound, and the kind of product that you are looking for, the kind of APIs that you are looking for. It can be streamlined and focused. And finally, out of these 234, only 15 were the one which were taken for further analysis. So this reduced the time which was there for isolation of new compounds from a particular species. Otherwise, one compound, if you screen them, it will take so much of time. But because of this high resolution technique, by combining the sophisticated techniques, you can always go for it. So dereplication is one of the important points in this. And also, I will just skip this right now. There are many so genome mining is also one of the methods by which the bioactive compounds which are isolated from marine organisms, they can, by applying this method, it will lead to characterization of natural products. For example, it has been shown that the bioactive compounds from a particular sponge, you are able to characterize using single cell genomics, led to the discovery of gene clusters. And they are very good source for anti-cancer agents. Genetic engineering can be applied. So right now, because of time, I will just go into the challenges again. Maybe I'll share the, you can share this presentation. It is all like we have given the reference from which we have, taken the matter. So definitely, many industries, they're collaborating with our, academia, so that they have a research base for, for whatever product they are getting into the market. It is much more authenticated. So generally, going through the challenges once again before concluding, in general, can the constituent international and regional scientific groups collaborate and gain a voice to promote the importance of natural products and their roles in society, in general? And can IPRs, conflicts between CBD protocol and TRIPS agreement, be resolved to enhance investment in natural product development? These are all the challenges in natural drugs that you see. Then there is sustainability issue. Can all the medicines be regarded as sustainable commodity and the needs of 2030 be met with sustainable sourcing? Because any kind of source that you're utilizing, yeah, most of the synthesis that is being done is based on the natural products which are being isolated. Yeah, so the impact of climate change on the accessibility of traditional medicine be assessed. So this all is providing an opportunity, to check the impact of climate change on the accessibility of the traditional medicines. Yeah. Um, then can traditional medicine be developed to become consistent, safe, and effective? So consistency is very important, and the efficacy should not change. And can the translation of meaningful research results on traditional medicines and phytotherapeuticals to product and practice be made more effective for patient benefit? Yeah, this is one of the challenge. Then when we are speaking about uh, those traditional medicines, they can they be sustainably developed and identified and conserved? This remains a challenge, and global harmonization of the diverse regulations becomes a reality, so that it gives an opportunity. And when we are talking about technology which actually is translating what we are doing at the lab scale level in our own areas, yeah, whether as a student, as a researcher, can the important emerging spectroscopic, chromatographic, remote sensing, and deep learning technologies be continuously integrated into the natural product sciences? And can the databases which is being provided by only by one industry right now, so can we have many libraries which, if you name a drug, it is already there? So though there are chemical libraries, but a thorough library for as such dedicated to natural drugs the complete secondary metabolic profile of a plant be determined. Yeah, these are all the challenges which we see in then plant analysis. Can the extraction separation, what we were, what the paper was shared in 2021 about the bacterial, about the analysis part, the purification, extraction, the problem that a person faces because of the less the less yield of a particular API that we are looking for, or the natural products. So can there be an extraction, separation, purification, identification can be done in the most sustainable technologies? Yeah. And a database for DNA barcodes for all traditional medicines be achieved. So all these challenges actually, in synthesis also, um, can vegetables and other sustainable and recyclable natural diverse reagent become an integral aspect of production of both drugs and chemicals? And the complete synthesis of complex drug molecule, um, can the complete synthesis be achieved using only recyclable or renewable reagents? If we can do that, which is from utilizing drugs from agri horticulture wastes, like we had isolated carotenoids from agri waste. We have developed a technology for the same, which is also being patented. Yeah. So can we do that for all the major drugs which are used in healthcare system? This will also reduce, this will also uh, help in utilizing the sources in a greater way, all our sources. Then can the reliance for profit scientific journals for the distribution of scientific information be significantly diminished? So we know there are many predatory journals, and recently a report came on how people, there were 80 research papers by DRDO scientists which have been found where there is copying of image. Yeah. And and there are many journals which claim to be in Scopus, but are they are not actually present in Scopus. So the reliability of the data should be there. So whatever information which we are getting, the challenge remains, because not every information should be published. Only when it is thoroughly reviewed by peer reviewed journals, it should be published in any journal, so that the authenticity remains. People who do research based on this, or the future of science also depends upon the kind of data which is being published in the public domain. So there are opportunities present in all these challenges. There are many challenges which are there. These are few challenges which I have told you, and the few opportunities which I could bring out of the papers that had gone through, and also by the work done by me, like how I am looking forward to doing more research in my area. What are the gaps? So we have to look for the gaps. In the gaps lies the opportunities, and in the gaps there are challenges which we have to actually address. And only when we address, whether it's in any area, so how can I utilize the knowledge that I have, mm, by combining, by going for combinatorial kind of research, because it cannot be done alone. One person cannot do everything. So we need different colleagues who can work together in synergy, and different streams which are required. So here with this I'll end, because I think I've short up time. That's why I skipped few slides, but I can share those slides. Yeah. So thank you very much.
Thank you, madam. Thank for this wonderful brainstorming section on drug discovery. You have covered, I think, almost all the topics, which not only include challenges and opportunities in natural drugs, but you have also given insight of drug discovery, patenting, as well as the current scenario of herbal drugs in India. With this, I request the delegates, if any one of you, if any of you have any questions, you can please unmute your mics and ask.
Hello, can you hear me?
Yeah.
Yes, I can hear you.
Actually, thank you, ma'am, for your presentation. Actually, I would like to know, you mentioned about one drug, like, somebody is suffering from kidney failure, and like with one kidney, that person is living. Yeah. So I would like to know the herb,
So in this, actually, that the therapy that I was talking about, it's a small magnet therapy, and it's a very effective technique which requires a simple blood test, and then you have to put those magnets in your hand, and actually the points in which the pressure is being held, it maintains the toxicity of the body. So that that friend of mine who is there with one kidney is surviving for almost 20 years that she's with one kidney. So I will pass on the information of that doctor to Dr. Pasha, and I think from there you can then take the information. He's known in AIIMS in Safdarjung, and he has treated many patients with different kind of ailments. So for kidney, I think that will be the best part, because it really helps you to reduce the toxicity and revive, because he's such a thorough person that I came across, that there's a high court judge whose wife had a neurological disorder. So the doctor had given 800 grams of medication every day, which was a very, very high dose. And both her father as well as brother were doctors. But after undergoing this kind of, like, I'm not telling that every kind of therapy is effective, but after, he could reverse it, he could reverse it. She's thoroughly fine. So whenever he comes, he actually in Noida, he sits in sector 50 at their bungalow only. He checks everyone. So they have dedicated it to, him.
Okay.
Yes.
Thank
So
you, ma'am. Thank
I'll
you very
send
much.
the address.
Yeah.
Please get in touch with him, and
Okay.
it's very effective. I have seen
Okay.
it in. Yeah, I'm very thankful to that friend. Actually.
Okay. Thank you very much, ma'am.
Yeah, and somebody has asked what are the opportunities for establishing evidence for COVID treatment using natural product. So, what are the opportunities? There are plenty of opportunities, what we had shown for COVID treatment, like by utilizing our traditional knowledge. How can you use by using those drugs which have been used for SARS? They by seeing the repurposing of drugs also, which have been used as antiviral drugs, we can use, and especially the formulation by, I have got a kit of All India Institute of Ayurveda, which I had kept in office. So they have this Swasth Rakshak also. In this, they have used all the medicinal plants like Embolica, gingiber, and they have used Terminalia bellerica, cinnamon, they have used dalchini also, Glycyrrhiza glabra, and Piper longum, as well as Piper nigrum, and Ajmoda, that is apium, and lavanga, which is Sizum aromaticum. And this actually treated many of the defense people. That was one of the centers of COVID. So maybe, by taking the lead from already made formulations against immunity, which have been used, like we had popular Tulsi Kada, we had popular Kadas at that time. People were, and still they are drinking many of them for immunomodulation. We can utilize the many opportunities, because we have literature backup, we have evidences in if you go for observational therapeutics, we have evidences for telling that they have antiviral action, and then taking the leads from there, especially for cough and cold, for congestion. There is an opportunity in COVID. There is plenty of opportunities, so much of, and it starts with, or you can target the side effects. How does it start? Like in many cases, it is starting with diarrhea also. There is high fever. Yeah. So that high fever is not coming down. Yeah. Then there is lack of immunity. It is attacking the heart. Yeah. There is swelling of legs. I personally had a lot of swelling in my legs. So the areas, the side effects which a person, there is the hair growth stops, people who are taking remdesivir. So either you go into the side effects and cure them, or you can go straight away for congestion, or you can go reverse, in the reverse manner, by when the symptoms starts, you can target the symptoms. Yeah, if there's diarrhea, you can target diarrhea. If you can control at that time, yeah, it will not aggravate like that. Yeah.
Thank you, madam. Any other speaker?
Yes,
Dr. Shankar.
Yeah, yeah. Good afternoon to everyone. I thank KLE College of Pharmacy for conducting such a great FDP session for all the faculties. And it absolutely, I'm listening to all the presentations since day one, and all are thought-provoking sessions. And today I'm very impressed with the Madam Harsha Kharkwal Madam's presentation as well. And this is Dr. Shankar from JSS College.
Thank you. Thank you, sir.
Yeah, working as assistant professor in Department of Pharmacognosy. My area of research is nano-herbal technology.
Okay.
So, yeah, as a part of that, I would like to ask you, madam, is there any, any product that has been, marketed using silver nanoparticles? As you mentioned, few natural product mediated silver nanoparticles have been prepared. So, yeah. Is it efficaciously used for the treatment of any ailments in current scenario?
So we have in general seen the effect for antibacterial activity, and the talks are going on with industry. As such, we have not marketed, but there are already many formulations which are present in the market, which are nano based. And especially, I think when we go for silver nanoparticle, silver has antibacterial properties. So
Exactly.
silver nano formulations we have utilized for our medicinal plant in in sunscreen cream, and also it has got very good photocatalytic activity. If you if you actually collaborate with any nanotechnologist, you will see that it has got very good photocatalytic activity, as well as there are papers which tell you that it will enhance the solubility of the drug. So especially in case of curcumin, to enhance the bioavailability, we can go for that kind of research also. So
Yes, great, madam. So we are working on Alzheimer's as well as diabetes, insulin receptor sensitization using silver as well as selenium nanoparticles, utilizing our kingdom of natural products. So we are utilizing natural products and connecting them into nano, and applying them into different kind of drug delivery systems, such as bioavailability increase, as you said, absorption also we are increasing. So as you said, silver nano, so it is absolutely nowadays we are trying to get some patents also on that.
Okay, great.
Thank you, madam, for your thought-provoking session as well. Thanks a lot.
Thank you.
And thanks to the organizing secretaries and the principal of our KLE College of Pharmacy.
KLE College for Pharmacy.
Exactly.
And I must thank Kulkarni sir also.
Yeah.
He's the one.
Okay, madam. Thanks a lot for the information.
Yeah, and Dr. Pasha for all the coordination, the principal and everyone present there for patient listening. And I know that you all all of you who have joined are the faculties who are already knowing many things. But I feel that when we share our information and we share the knowledge that we have or the work done by us, something new definitely comes up.
Yes, madam.
And that is the purpose of FDP, because faculty development somewhere, maybe I feel that everyone knows maybe that part, but maybe by speaking out and reaching out to others, how did I succeed in a particular way? So maybe somebody can get it, even if out of the people, so many people who are attending, even if I think 4 or 5 people get some novel research idea out of this, then I think it's a success to actually for any FDP. If anybody gets some lead, some kind of lead from any kind of lecture which is being delivered. So I'm very, very thankful to KLE College of Pharmacy for organizing this and inviting me.
Madam, we have a few more
just I wanted some information, madam, from your side. That's why you talked about this library, extraction library.
Just,
will you highlight it, madam? Just I wanted some information. That's what is the avenue for this to approach this library?
Yeah,
how it can help.
Yeah, so I think for extraction library, in Anchrome, we have libraries for the extracts for phytoconstituents, like that, in industry. In our case, the extraction protocols, with extraction protocols of different sources that we are utilizing, whether it is of medicinal plants or marine drugs or animal sources or any kind of sources. Most of us, we are utilizing medicinal plant only. So the standard protocol procedures, with that, a library can be established. Otherwise, every industry has got their own SOPs. And with that SOPs only, mostly we get that percentage of extract. Like in case of Alzheimer in mucuna, we have a senior scientist here, Joshi sir, who works on that. So he's developed a technology where 40%, it is used against, and which is 40% levodopa extract is used for curing this Alzheimer, and which is more effective than the synthetic one. But the procedure, and the exact way it has to be done. Yeah, it is there in one of the SOPs booklet, which is there. So a library can be made. Like only one industry, Nicholas Pirmal, is right now having that kind of library, but it depends upon only when we collaborate with some kind of industry, then we can get access to these kind of libraries. Yeah.
Thank you. Thank you.
Yeah.
Good afternoon, madam.
Excuse me, I have something to ask. For example, madam, here in terms of academic institution, do you feel there is a chance for developing such library over may not be immediately over a period of time?
Yes,
academic
yes, sir.
Institutes can also develop their own libraries, which may be shared among other institutes.
Other institutes,
the process becomes easy.
Yes, sir, and we can get grants from National Medicinal Plant Board. NMPB, yeah, we will get grant for this kind of libraries, because NMPB is really promoting, this kind of research where, because it gives so much of subsidy to farmers for cultivation, for processing, for development, and if and the main part is like whatever we are importing or exporting, if we have the the library for the extracts, it will enhance the export value also. So we are right now number 6 according to NMPB data, but then we can rise up to somewhere near 1 also, if we have, if we can all join hands and whatever, because not everyone is working on all the medicinal plants. So we can do a collaborative research in which we can take industries as well as few people who are working in this kind of this field, and then together we can develop a library. That will be that is a very good thought. So maybe from Nicholas Pirmal only we can join hands. That is not an issue. We can always join hands with them.
Or we can have a consortium of different academic institutes to develop a combined library.
Yes, sir. Yes, sir. That will be a very brilliant. It's a brilliant thought, and we can go about it.
Thank you, madam. It was informative lecture.
Thank you,
sir. I'm thankful to you for this.
Sir, I'm also thankful to you. Yes.
Yeah. Thank you. I thank Giriraj sir also for giving us such an excellent, eminent resource person for this FDP. Actually, sir recommended your name, ma'am, and you are here today, and your presentation was indeed very, very excellent, ma'am. Ma'am, one question from my side. We are working on mastitis, a veterinary product. Recently, we have started working on this. And problem what I am facing is I take an extract, my formulation is not that good. So I moved into a phytoconstituent now, pure one I have taken and I have started, but the cost is very high. The cost is very high. So what suggestions would you give me for this type of a formulation to bring into market? I'm not finding a way. The cost,
whatever activity that particular constituent is showing on the animal, like you can always get in touch with IVRA, also Indian Veterinary Research Institute, and I have I also know few of the scientists there. So whatever activity, like I always tell the students, look out for the chemical moiety which is responsible for a particular action. Yeah, if that particular chemical moiety or that particular group which is responsible for a particular activity, whether it is antibacterial, antiviral, analgesic, or any kind of pain that you're addressing to, or any kind of tumor or anything, so you will have you have to just check whether any other phytoconstituent has the same property, and then just compare the cost if it can be reduced, but the therapeutic activity should not be compromised with, and then you can go for that. The other part is that if only one particular constituent is showing the activity, which part of that particular plant So you can do a comparison with the extract, or you can combine the drug that is already there in the market with this phytoconstituent to reduce the cost, as well as to enhance the therapeutic activity. Yeah, because one is cost reduction also. It can be either way. The drug which is already there in the market might be more costly than what you're preparing. So maybe combining both of them, but you have to see if there is any drug to drug interaction or if there's any toxicity. If there's no toxicity, then you can just go about it. So there are maybe we can chat over the mail also, and you can send me and we can see what are the steps that you can do so that you come to a cost effective formulation. And in your formulation, if you are getting any difficulty, I think the formulation can be corrected now.
Yes, ma'am. Yes, ma'am. Sure.
It
Thank you
can
so
be-
much, ma'am.
Yeah, yeah. Thank
It
you.
was really a very informative and nice interacting with you.
Thank you. Thank you.
On behalf of Kaili College of Pharmacy, Hubli, and on myself, on my behalf, I thank Giriraj sir for liaising and connecting us to Harsha madam. It was a very informative session. Thank you, sir. Thank you very much. So with this, we come to an end of this session. I request Aki madam to conclude the session.
Thank you, madam, for your excellent presentation, where you have provided key strengths of Indian pharmaceutical industries along with the detailed insights of opportunities and challenges in natural product discovery. I thank you once again, madam.
Thank you, ma'am. Thank you so much.
Thank you, sir.
Everyone.
Thank you, madam.
Thank you.
Yeah.
Thank you.
Yeah. The next session we shall begin at 3 p.m. 2:45. I request you all to join. The speaker for the session is Vadiraj Bharadwaj, formulation consultant, Injex Labs Private Limited, Bangalore. His topic is new product development inspired by Ayurveda.
Thank you, sir.
Yeah.
Thank you, everyone. Thank you.
Thank you.
Thank you.